So this part I'm going to try and summarize the phases of mitosis for us. So interphase, prophase, prometaphase are the first three, and then we'll move on to the last three as we go into metaphase, anaphase, telophase, cytokinesis. So I like some pictures. I've tried to summarize the pictures in words that follow these. So in interphase, there's some key things happening. I'm just going to talk through the diagrams, and then you can read through the words if you like. So interphase is the biggest part of that cell cycle. This is where we have the chromatin, or the chromosomes are going to double. It is diffuse, we'd call it chromatin here. In interphase, you can still see the nucleolus, and you can still see the nuclear envelope clearly intact. Everything has doubled in interphase. In addition, or along with that, are the centrosomes, and each centrosome has a centriole pair. So this is sort of the normal state of life for the cell. And after that, we're going to move into prophase. So prophase, what's going to happen is the chromosomes are going to coil and they become visible. So we've got the two sister chromatids joined at their centromeres. And you can see here, they're sort of like, if they were an X, their arms and legs or the tops and bottoms are together, but that would be the centromere in the middle of them, and then the tops and bottoms. So the other thing that's going to happen here is the centrosome is going to send out the spindle fiber, or the mitotic spindle. After that, I put this in red only because not all publications list prometaphase, so it's somewhere between prophase and metaphase. The key things that are happening here is the nuclear envelope is going to break down. It has to get out of the way so we can make two new nucleuses, or nuclei I should say, out of this. And so what's happening here is the spindle fibers are elongating. The centrosomes have moved to the poles of the nucleus, and then we've got one more word, and of course it starts with a k sound. These are little motor proteins. They're called kinetochores, and those kinetochores line up facing each of the centrosomes, and it's where the spindle fibers are going to attach to the centromeres of the sister chromatids. So if you follow through the slides, most of the things that I've said should be there as the written examples of what's going on in each stage. When I move on from that, I can do the last stages. So metaphase, I like to think meta or middle. What happens here is that all the chromosomes have aligned in the middle or along the metaphase plate, sometimes called the equator, as these were called the poles. The spindle fibers are fully formed and have aligned. So those kinetochores, those motor proteins, have made sure that those double chromosomes are lined up directly in the middle so they can be divided equally. Anaphase, what's going on here is the cell's going to elongate, and the sister chromatids are pulled apart. They're separated. I always like to think of the shape of them as making kind of A's here, and that's how I remember what's going on in anaphase. But we would now call those sister chromatids now daughter chromosomes. The last one, this telophase and cytokinesis, don't call it telekinesis. I had somebody once make that mistake, and it was super funny because if you think of what telekinesis is, it's moving things with your mind. That is not part of cell division. Telophase and cytokinesis often overlap. They happen pretty quickly together, but they are two distinct words in the world of biology. And so basically everything that's happening in telophase is the reverse of prophase. And so we're going to have the nucleolus coming back. We're going to have the nuclear envelope coming back. We're going to have the everything sort of coming apart into two nuclear regions or new nuclei. The other thing in cytokinesis is that separation of the cytoplasms, the separation of the cells, exactly. And these are different in animal cells and in plant cells. So the next slides are basically what I said there. So metaphase, think middle, ana, they're separating and going back. The cell gets longer. Telophase is basically all the reverse of prophase, right? down there. So if you've understood prophase, then you just have to reverse that for telophase. And cytokinesis is when the cytoplasm actually divides into separate cells. So in animal cells, we get a cleavage furrow where it's just the cell membrane or the plasma membrane coming in on itself. And in plant cells, we have both a plasma membrane and a cell wall. And so what forms in plants is a cell wall. We have vesicles depositing materials for a cell wall in the middle, and that cell plate grows out. It sort of hovers in the middle and then extends to the edges, dividing the contacts of the two cells. And so both of the cells have plasma membrane, and a cell wall in the new cells that are formed. These are images of both. So these would be animal cells over here. And this would be a schematic of plant cells. And so here you can see the cell wall all the way around. And this section in the middle is the cell plate being formed. So it is a bit of a shadowy line, different than the schematic, but under our microscopes, you can see those things. The control of mitosis is important, and then there's several factors that control cell division. And so that would be nutrients. We need enough. We need growth factors, proteins, enzymes, those sorts of other things that stimulate division. And then also there's some other inhibitions, so density-dependent inhibitions. If I have a cut on my finger, I want the mitosis to stop before I overgrow those skin cells. The other thing is anchorage dependent. So if I skin my knee or some cells of me are not attached, I'm not going to regrow a person from that. So those cells need to be anchored to the rest of you in order for cell division to happen. And really normal growth and development is contingent on the control of cell division and the regulation of the cell cycle. So the cell cycle is actually got some bailout points. It's got some fuses built into it so that if something is wrong, then it's going to stop the cell cycle and the cell won't divide. And so basically there's some key checkpoints here that if something is wrong with the cell, then it's not going to continue on. It's going to get sent out and you won't have more cells like that cell dividing. They're basically stop-go signals that regulate or growth factors in the main signals. If we were to talk about mitosis out of control, so too much mitosis, that would really be at the root of how cancer works in your body. So normal cells can convert to cancerous cells if there's a mutation, maybe due to any environmental factors or sometimes genetic factors. And basically these cells escape those regulations of the cell cycle. So they don't respond to those normal regulation signals. They blow the red light. And so cancer cells going to divide rapidly. Often they don't even need the growth factors, and they can spread to other tissues through the immune system. This is, how cancer can be so difficult. It's unrecognized by your immune system. Your immune system still sees those cells as you, so it doesn't get rid of them. And so that's why cancer can be tricky. Growth is not inhibited by other cells, and so you don't have that sort of check and balance, and the overproduction of cells is what we would call a tumor. You could have a benign tumor that stays where it starts, or that behavior can spread. You could have a malignant tumor that spreads by metastasis, or it metastasizes in your body. So these last two slides are here just as a bit of a teaser to part B. So everything in part A, we've been talking about mitosis, and in part B, we're going to go on and talk about meiosis. Now, meiosis is different in a lot of ways, but it is also the same. We'll go over some of the key vocabulary, and this slide is good. I think I may end up using it when I teach part B as well, because there's a lot of key differences illustrated in here. You can see that we have one parent cell. In humans, that would be 46 chromosomes. It would be a diploid cell. If we go through doing what we just did, we can see that the chromosomes line up as double chromosomes in single file. And then the sister chromatids separate. So over here, if we count the number of chromosomes, we would still have, whoops, sorry, wrong number, 46 chromosomes. And this is what we want if we're going to grow or repair our cells, or if we're a paramecium or an amoeba, a single-celled organism. So these down here would be diploid. You can see that we make 2 cells out of this, right? And these cells would be identical to the parents. So daughter cells, they'd be same as parents. If I were to go through and talk about meiosis, I'll switch colors up a little bit. So meiosis, we could start with a diploid cell, but we're going to make haploid offspring. In the world of humans, that's 23 chromosomes. So these would be gametes. Sometimes we call them sex cells. They would be sperm and egg in humans. And some key differences here is that we're not going to go through it once, but we're actually going to have 2 cell divisions, so 2 rounds of cell division. The thing that happens that's the most significant is this reduction in the number of chromosomes, right? And that reduction happens right here. So when the chromosomes line up here, they line up as pairs. Those pairs have names. They could be called homologous chromosomes, or they could be called tetrads. Usually I like the word tetrads because it's much shorter. And what happens here is instead of the sister chromatids separate, the homologous pairs separate. And this is really the key thing that reduces the number of chromosomes. So we'll talk more about meiosis in the next part of chapter 8. The other fancy word, this is gametes over here. These would be somatic cells. If they are regular body cells, like most of our cells are diploid. And we'll carry on with talking about meiosis in the second part of chapter 8. I also put this in as a place that kind of on one slide compared and contrast mitosis and meiosis. So that might be helpful as you're kind of looking forward to the next part of chapter 8.